metabolism of high density lipoprotein. Okay. Synthesis of high density lipoprotein started in liver and intestine. Okay. Number one, synthesis or biosynthesis or gene expression of alpha 1. Alpha 1 gene expression intestine and liver. Okay, so alpha 1 will be secreted. Alpha 1 or A1 protein out A1 from intestine and liver. Epithelial cells or enterocytes to the blood. Okay, so A1 will be secreted to the circulatory system. Blood. A1. Okay? A1. A1 is the activator for eliciting cholesterol acyltransferase. Okay? So alpha 1 is activator for eliciting cholesterol acyltransferase. Okay? There is phospholipid in the blood. Okay? So phospholipid will bind. Okay? Will form discoid high density lipoprotein. So this is the phospholipid. A1 eliciting cholesterol acyltransferase will bind with the A1 activator. Okay. This is discoid high density lipoprotein. The discoid high density lipoprotein is hungry lipid. So the cholesterol in the blood from low density lipoprotein and from epithelial cells. From cells. Okay. Okay. This is the cell membrane of the peripheral cells. The A1 second mechanism, A1 free in the blood, alpha 1. Alpha 1 love the cholesterol. This is cholesterol inside cell membrane. When the A1 near the cell membrane, يقترب من الcell membrane, the cell membrane will be changed in configuration. Will it change it in configuration? Okay, so the A1 will bind with the receptor. This receptor, we called it ATB binding cassette protein. ATP binding cassette protein. So the A1 will near the channels with the helping of ATP to ADP. Okay, lots of ATP will be consumed, changing the shape. Changing in shape, dilation will occur. So dilation. Then cholesterol can be released from from the cytoplasm to the A1. Okay? The cholesterol plus A1 plus phospholipid, as in this case, will form a very high density lipoprotein. So, very high density lipoprotein, cholesterol, phospholipid, cholesterol, LCAT, eliciting cholesterol aside transferase. Okay, the cholesterol will be acylated. By phospholipid. Tarana composition of phospholipid will involve in esterification of cholesterol. So cholesterol plus phospholipid, this is phospholipid, lecithin cholesterol aside transferase, cholesterol ester plus lysophospholipid. So again, the synthesis of high density lipoprotein is started in liver and intestine. Hypolipidated, no lipid, A1. A1, there is a receptor in peripheral tissues. Okay? Receptor in peripheral tissues. Okay. Interaction, conformation, interaction with the ATP binding cassette protein, dilation, the cholesterol will be released in circulatory system, bind with A1 activator for LCAT with phospholipid in the blood will form discoid. Okay. Hypolipidated. Why? Because cholesterol not in esterified form. So cholesterol will be esterified to cholesterol ester. Okay. Again, the discoid and cell membrane. Okay. So there is two mechanisms by which cholesterol can be transported from cytoplasm to the circulatory system. Number one, by ATP binding cassette protein. Number two, by scavenger receptor. So the scavenger receptor, there is no ATP used for transportation of cholesterol. Scavenger receptor. From scavenger receptor, the cholesterol will be transported from tissues to the pre-high density lipoprotein. Okay, so pre-high density lipoprotein. Okay, cholesterol ester, cholesterol. صار كل كوليسترول يتحول إلى كوليسترول إستر يعني low free cholesterol concentration or low free cholesterol concentration. So cholesterol will be transported again. LCAT A1 cholesterol transported. So the pre-high density lipoprotein will convert it to high density lipoprotein A3. What is the difference between high density lipoprotein 3 and very high density lipoprotein? It is just in content of cholesterol. Okay? So more lipidated. Okay, again, the high density lipoprotein also by scavenger receptor will convert it to high density lipoprotein 2. High density lipoprotein 2. Difference is only in content of cholesterol. Okay. In liver, there is hepatic lipase. And hepatic lipase work as ligand between hepatocytes and A1. This is A1. Okay? So they يشتغل as a ligand and also as the enzyme. Ligand and enzyme. Okay. And also here there is a scavenger receptor. Scavenger receptor. Okay, scavenger receptor صار عندنا two roles. Number one, transportation of cholesterol from peripheral tissues to the high density lipoprotein. Number two, absorption of high density lipoprotein 2 by liver. Okay, another high density lipoprotein 2, the content of cholesterol more than high density lipoprotein 3. Okay. Okay. The all content of high density lipoprotein 2 will be absorbed except A1. There are two pathways, and when all content كل high density lipoprotein 2 absorbed, only A1 back to circulatory system. Okay? Number two, second probability, الكوليسترول ester will transported and high density lipoprotein 2 will converted to high density lipoprotein 3. Again, cholesterol transported, high density lipoprotein 3 converted to high density lipoprotein 2. Absorption. صار هنا في two احتمالات. Number one, all the content will be absorbed except A1. Okay? The second choice, the cholesterol ester will be transported and the high density lipoprotein 2 converted to high density lipoprotein 3, where is low content of cholesterol. Okay, the synthesis of the high density lipoprotein started in intestine and liver. Also, this C1, C2, C3, A2 release it with A1. Okay, but they are not the main composition of the high density lipoprotein. So high density lipoprotein to be formed must be A1. Structural function. These lipoproteins just involved in metabolism of other lipoproteins. Activation of lipoprotein lipase, conversion of nascent to remnant chylomicra, or very low to intermediate and low density lipoprotein. But structural function, A1. So synthesis started with the releasing of A1. A1 love cholesterol will bind with the cassette receptor, ATD, conformation, releasing of cholesterol, bind, listing cholesterol acyl transferase circulating in the blood, bind with A1 activator, lecithin will involve in esterification of cholesterol to cholesterol ester. Hungary again with cholesterol تحول إلى cholesterol ester. That means the free cholesterol very low. Again, scavenger receptor releasing of cholesterol from the cytoplasm, cytoplasm of the peripheral cells to the high density lipoprotein. Okay, pre-high density lipoprotein converted to high density lipoprotein 3. Again, scavenger receptor. Again, release it to high density lipoprotein 3, high density lipoprotein 2. The highest lipidated high density lipoprotein is high density lipoprotein 2 by scavenger receptor. So scavenger receptor in liver absorption and peripheral tissues is the mechanism by which the cholesterol transported to high density, pre-high density lipoprotein. Okay. So this is the high density lipoprotein metabolism. High density lipoprotein. كيف؟
مش في انه قلت في خيارين انه يا اما التراي اسايل جليسرول وبعدين نقول دي ال 2 ولا يا اما بنخلي الاي في خيار ثالث انه كلها على بعض
لا ال A1 cannot be absorbed. A1 cannot be absorbed. Okay. Alpha 1 cannot be absorbed. هذا circulating. Okay. Okay. So this is the mechanism of high density lipoprotein. There is a high density lipoprotein cycle. What is the high density lipoprotein cycle? It is conversion of high density lipoprotein 3 to high density lipoprotein 2. High density lipoprotein 2 absorbed by scavenger receptor in liver. High density lipoprotein 2 converted to high density lipoprotein 3. This is the high density lipoprotein cycle. I want to complete two probabilities. Complete absorption except A1 or only transportation of high cholesterol ester to the liver. When the cholesterol ester only transported, that means the high density lipoprotein 2 will be converted to high density lipoprotein 3, the less lipidated high density lipoprotein. So the synthesis started in liver and intestine. Apoprotein C, apoprotein E, apoprotein A, كلها in liver.
يخدم كإنزيم يخدم كليجند في أي واحد يخدم كليجند.
هو موجود على السكافنجر ريسبتور هنا. هذا السكافنجر ريسبتور هو هنا على السيرفس أو السيل ممبرين فيساعد على الانتر اكشن.
الرياكشن يخدم كان
ليجند. نعم هذا ليش نقول مثلا هاي density lipoprotein 2 ممكن يتحول إلى 3 without absorption. ليش؟ ليجند هيك at the same time هذا cholesterol ester by hepatic lipase to cholesterol and fatty acid. Okay, so هو هيعمل hydrolysis even to cholesterol ester. So كل الcontent of high density lipoprotein 2 can be hydrolyzed by hepatic lipase, except of course the proteins C2, C3, E. Okay, there's lysosomes, peptidases enzymes. Okay, so high density lipoprotein 2 started in the liver and intestine. So the function of high density lipoprotein, it is not only transportation of cholesterol from peripheral tissues to the liver, also involved in metabolism of other lipoproteins. Without high density lipoprotein, the very low density lipoprotein cannot be converted to intermediate density lipoprotein and low density lipoprotein. Also, nascent chylomicron cannot be converted to remnant chylomicron. Okay? Involved in metabolism of other lipoproteins. Okay. The high density lipoprotein 3. This is the low density lipoprotein or chylomicron. The cholesterol from low density lipoprotein or from very low density lipoprotein can be transported cholesterol to the high density lipoprotein 3 or to pre-high density lipoprotein by aside transfer protein. Aside transfer protein. So aside transfer protein. Sulfate protein can in the blood, in the plasma, فهذا low density to the peripheral tissues. Yes, ممكن في الطريق هكذا. الكوليسترول transported from low density to high density by acyl transfer protein. Acyl transfer protein. So acyl transfer protein will transport the cholesterol, ester, and cholesterol to the high density lipoprotein and back to the blood. Therefore, the high density lipoprotein is good. Why? Because it's the lipoprotein responsible for clearing the cholesterol ester from the blood. So the cholesterol back to the liver. Good. كما قلنا الكوليسترول بدينا انه يكون موجود وين؟ الليفر. والباد انه يكون موجود في البلد. Why? Because high probability for aggregation of high low density lipoprotein, and low density lipoprotein will be aggregated where? In wall of blood vessels, atherosclerosis. Okay? reverse cholesterol transport هذا بنسموه فيها ميكانيزم reverse cholesterol transport. reverse يعني هو عادة ينتقل من الليفر إلى البريفرال تيشو. reversion rebacking of cholesterol from peripheral tissues from low density, high density, from low density, intermediate, chylomicron, low density to the high density lipoprotein back to the liver. Okay. So cholesterol flux is brought about by esterification of cholesterol under the effect of listed cholesterol aside transferase. So okay. So cholesterol ester, eliciting cholesterol ester aside transferase is the key point in metabolism or transportation of cholesterol from peripheral tissue to the liver. إذا كان هذا ما يكونش active, the cholesterol cannot be esterified. إذا كان ما يكونش esterified, شو يعني ذلك؟ scavenger receptor cannot be active, cannot be transported. لأن الترانسبورتيشن ما تعرفوا أنتم في transportation. عادة الترانسبورتيشن يعني من الهاي إلى اللو صح؟ من الهاي إلى اللو. لما الكوليسترول يتحول إلى كوليسترول إستر، إيش يعني؟ معناها تعني لو فري كوليسترول. معناها الكوليسترول will transport from cytoplasm to the lower concentration. جوا؟ كوليسترول تحول إلى كوليسترول إستر. الكوليسترول يعني free cholesterol, low free cholesterol. Low concentration of free cholesterol, high concentration of free cholesterol. So free cholesterol is transported. So without esterification, the cholesterol content will be high. then the scavenger receptor cannot work. And I've gone high on a high, cannot be transported. Okay, so this is the cholesterol aside transferase. Also is K volume in synthesis of high density. So the very K volume أول نقطة ضروري يصير ريليزنج للألفا لايبوبروتين أو بروتين A1. إذا كان ما يتكونش ال A1 hyper cholesterol. Why? Because the only mechanism by which the cholesterol reverses to the liver is high density lipoprotein. ولهذا الهاي density lipoprotein كل ما نشوفه في التحليل هاي كل ما هو ضد. يعني كان كل ما نشوفوه more than 40 ملغ بر ديسيليتر. Very good. لو كان يقل كان مثلا 20 أو 15 نقول له شنو خشينا في الريسك فاكتور. الريسك فاكتور يعني الريسك فاكتور يعني معناه فيه حيكون probability أكثر لإنه يكون فري كوليسترول إنه أكثر. لأن من هنا هكذا ينتقل السايد ترانسفيرز مع الكوليسترول. كل ما بيصير كوندنسيشن في البريفرال تيشوز للكوليسترول استر حيصير الهاردنس of the cell membrane rigid. Yes. البلاد فيزلز حتبدا ريجيد كأنها مش فليكسبل ريجيد يعني كأنها تنكسر ما هياش مرنة. اوكي rigidity of blood vessels. It's bad. Okay, scavenger receptor B1 and في lots of scavenger receptor in our biological system. هذا حناخدوها إن شاء الله في كورس الهرمونز classification of receptors. Okay, لأن في scavenger receptor B1, scavenger receptor B2. Okay. ATP binding cassette transporter A1. ATP binding cassette protein A1 plus A1 in this case. Okay, B1 A1. Okay. Okay. طبعا في mechanism by which the A1 or cholesterol can be transported by ATP binding cassette protein. أقرأها هكذا لكن molecular biology مش هنتطرقوا له في الامتحان الميكانيزم هذا بالتفصيل. اوكي نعرفه احنا بس بالبايوكيمستري. It is nearing A1 to ATP binding cassette protein. Conformation occur to the cell membrane, cytoplasm. Cholesterol can be open the channels and cholesterol transported to the A1. Okay. So cholesterol ester is the protein responsible for transportation of cholesterol from the chylomicron and from low density, from very low density to high density lipoprotein. Okay. What is the function of high density lipoprotein? Scavenging action. High density lipoprotein scavenges or return back from extra liver organism, liver back. Okay, involved in metabolism of chylomicron, very low density lipoprotein, intermediate density lipoprotein, by activation of lipoprotein lipase. Okay. High density lipoprotein stimulate prostacyclin synthesis by the endothelial cells. Prostacyclin, what is the prostacyclin? It is the cosanoid. Prostacyclin. Cosanoid. هي بروستاجلاندين واحدة منهم بروستاcyclin. Anti-inflammatory compounds. Anti-inflammatory compounds. Okay. Anti-inflammatory compounds also prevent the platelet segregation. Preventing the thrombo formation. يعني high density lipoprotein decreasing the probability of stroke or thrombosis formation. Thrombosis, جلطة دموية. Okay, so high density lipoprotein negating the prostacyclin. Okay, helps in removal of macrophages from the arterial wall. Okay. I think in the macrophages, immune system, yes? Immune system. By condensation of macrophages in the wall of blood vessels, okay, can lead to releasing of histamine, secretion of pro-inflammatory compounds. Pro-inflammatory, صح عند immune system. الماكروفاجز انوال blood vessels لما تتراكم هنا. Okay, the immune system can in any moment attacking the native cells. ماذا بيه تكون أنا in the human, not in contact with the wall of blood vessels. Okay. Okay, so it's a clearing factor. Helps in removal of macrophages from artery walls. فماذا بيه أنا macrophages الموجودة تكون مش on contact with blood vessels, with blood in the human circulatory system. Attacking pro-inflammatory reactions will occur. The pro-inflammatory reactions سبب dysfunctioning of epithelial cells. لما يصير dysfunctioning of epithelial cells ايش يحدث هنا؟ Transportation of low density lipoprotein under the cells, epithelial cells. Thromb formation. So يصير condensation under epithelial cells. Okay? Smoking also dysfunctioning the epithelial cells. Cigarette. Free radical dysfunctioning the epithelial cells. Homocysteine dysfunctioning the epithelial cells. So أي مادة تعمل dysfunction للepithelial cells يعني physiological function disoriented. So أي مادة تعمل attacking للepithelial cells. الepithelial cells هتفقد functions. هذا المقصود بها dysfunction epithelial cells. فشو بتصير هنا؟ بتزيد البرميابيليتي نفاذية بعض المواد للابيثيليال سيلز. وبالتالي تبدأ تتراكم under epithelial cells. يصير بروتين aggregation, lipid aggregation, cellular debris يعني حتى الخلايا الميتة أجزائها المتناثرة السيل ممبرين under epithelial cells تكون ترومب اتريوسكليروتيك بلاكيو، اتريوسكليروتيك بلاكيو، يعني هذه اتريوسكليروسس. صار الاتريوسكليروسس هذا مش فقط هاي دنسيتي اللو دنسيتي ليبوبروتين، اجريجيتد بروتين، فهو complex very rigid sometimes cannot be removed by surgery زي الواكس يعني removal ممكن يسبب hemorrhage. ممكن نعمل كان نستخدم القوة عنه ايش بيصير للسيل وول جزء منها هاي هذا very rigid. It is not only low density lipoprotein. Low density lipoprotein started aggregated. Okay, لكن في cellular debris، في albumin، في globular protein، كل يصير لها aggregation under the wall of blood. لكن هذا يحدث في خلال decades. Yes, decades. decades يعني إذا كان الشخص عنده هايبر كوليستروليميا مش في يوم وليلة حيحدث أتروسكليروسس.
قصدك عشان سريع
Yes. ممكن started from teenagers. Bad lifestyle. High carbohydrate intake. High lipid synthesis. Hypercholesterolemia. Bad lifestyle. Sleeping هذايا قصدي سهر Low synthesis of high density lipoprotein. صار لما يبدأ الشخص بهيك حياته نمط حياته ممكن يأتي في خلال عمره 40 سنة أو 45 سنة نلقى عنده أتروسكليروسس كارديوفاسكولار ديزيز. فكله يعتمد على اللايف ستايل. سبورت increasing the high density lipoprotein. High carbohydrate intake. More lipid. High cholesterol. High lipoprotein. Which one? Low density lipoprotein. With decades 10 سنوات اوكي مع smoking مع اوكي دو سبورت يسبب اثيروسكليروسس. اوكي
في نوعين من
LDL
A و B. قصدي مش شرط معناها لو LDL عالي معناها كوليسترول عالي معناها لا مش شرط.
ترى اعطينا مصدر معلومتك انت ما دام خشيت في شرط ومش شرط اكيد انت قاريتها في معلومه في كتاب مش الراي الفكري.